In this video, we're going to understand essentially everything we need to know about ribosomes. Ribosomes are composed of many polypeptides and ribosomal RNAs, which are held together by a massive quaternary structure. And this is, the quaternary structure is, remember we talked about quaternary structures, are structures with a lot of polypeptides that are coming together and interacting with one another. And that's what a ribosome really is. It's a very complicated structure. It's not pre-made like a RNA polymerase or a DNA polymerase. It's something like, for example, a spliceosome, which is made at spot. It's composed of different kinds of polypeptides and enzymes. In ribosomes, we notice that ribosomes will be floating in the cytoplasm, and the ribosome has a large subunit and a small subunit. And if you don't know anything about ribosomes, just know that ribosomes function are the primary key focus in translation, and they're reading the mRNA, and as a consequence, they'll be forming a polypeptide with the help of the tRNA. So let's quickly understand what's the difference between prokaryotes and eukaryotes real fast. If you're an IB student, they're just going to ask you what kind of ribosomes do you have inside a prokaryote, and the answer to that is 70S. If they ask you what kind of ribosome do we have inside a eukaryote, you'd say it's an 80S ribosome, and then you won't really understand why it's called why we have a S and what's an 80S and what's a 70S. And the answer to that question really just is that the S is a reference to the Sedberg's sedimentation rate. So the Sedberg's sedimentation rate is how quickly something will be sinking. Well, how quickly something will sink if we were to put it in a centrifuge. And centrifugation is a process when we have tubes that are spinning at a very high rate, and it causes the components inside the tube to sink at its rate a little faster. It's just fastening everything up in terms of sedimentation. So that's what the S just refers to, and the 70 and the 80S is referring to the components of the ribosomes. So let's talk about that in a little more detail later on, but let's focus on what the ribosome is in the first place. The ribosome has a large subunit and a small subunit. And inside the ribosome, we have three different sites. We have the A site and the P site and the E site. And your biology teacher might have told you the A site is where the tRNA comes into or attaches onto first, and the P site is where polymerization happens, or polypeptides polypeptides are formed. And then the E is where the tRNA falls off, but you never really understood why it's called the A, P, and E. And the reason it's called the A is because it's the amino acyl tRNA site. This is where the amino acyl first comes. An amino acyl tRNA site, tRNA is just a loaded tRNA. We've talked about this in the previous video. And then we have peptidyl tRNA site, which is, peptidyl just means that the peptide is growing. And then the exit site, E, stands for the exit site, where the tRNA is just falling off. So this is essentially, well, look away, you didn't see this. You're not seeing what I'm doing right now. Please notice that I didn't do that. So here's a question to ask. Now let's say that we have a polypeptide, a ribosome that's reading an amino acid. Now think about this. If it's going in this direction, now where do you think is the 5' and the 3' end? And if you saw it before, that's fine. Just think about it in this way. If it's a 5' being synthesized to a 3', we know that translation always happens in the 5' to the 3' direction. That's just how translation happens. So if that's the case, where is the 5' and 3' in this mRNA? So let's think about it. This is the mRNA. This is how it's being translated. So we say it's the 5' to the 3'. So look what's happening. Whoops, it's being translated. Ignore this right now. Just notice the movement of the mRNA. It's going in this fashion, and it makes sense because it's going from the A to the E. So look at that. So translation is always happening the 5' to the 3' end. So the 5' end has to be this end here, and the 3' has to be this end. So please memorize this, that everything, even transcription, is happening the 5' to the 3' end. That's very essential. So let's talk about the machinery behind the machinery that's needed for This is the machinery that's needed for translation, And you might have seen, okay, so we've talked about the difference of 70S and 80S ribosomes, and we've also talked about the Sedberg unit, which is the sedimentation rate, really. So prokaryotes have a 70S ribosome, and we have, for MCAT students, this is good to memorize because we will discuss what these different, or specifically the 23 S over here, and then we'll talk about this later on. But the prokaryote has the large subunit is the 50S subunit, and the smaller one is a 30S. And these are composed of other smaller enzymes and polypeptides. And remember that these sedimentation rates don't add up. So if we have these right over here are the components of the 50S large ribosome. We have a 23S and a 5S are ribosomal RNA, and we have 31 polypeptides. So 23 plus 5 is not 50. So don't be confused by this, by just adding up the ribosomal RNAs, because we have other factors inside as well, such as the 31 polypeptides and whatnot. Whoops, excuse me. My glasses just fell off in excitement. So we have 30S, and 30S is when we have 16S ribosomal RNAs, and we have 21 peptides inside it. So 21 peptides are a lot of peptides if you think about it. It just signifies, and we have 31 over here, which just really shows that these ribosomes are very, very complicated complexes. And let's talk about the 80S ribosome real fast. The 80S ribosome has a large subunit of 60S and a smaller of 40S, subunit of 40S. The three types of RNAs inside the eukaryotic ribosome, large ribosomal subunit, and that's the 5S, 5.8, and 28S. There are 46 peptides, and 31 polypeptides is a smaller one. 5S RNAs, 23. You can just see this. I don't have to speak it out. But the point is, you see that the ribosome of the eukaryote is bigger and is composed of more peptides and more RNAs. So it's a bigger complex. And we're gonna really quickly compare these real fast. So this is the components of the prokaryotes. So let's talk about the prokaryote first. And I forgot to write this here. So here is the prokaryote. So the prokaryote is going to have, this is really just summarizing what we talked about, the two kinds of our ribosomal RNAs with 5S and the 20S and 31 peptides, and that makes the 50S. And then we have 16 RNAs, and then we have 21 peptides, and that makes a smaller ribosomal subunit. And that just makes the entire 70S ribosome of the prokaryote. And this should make it all. This is just really recognizing that, we don't have ribosomes aren't like DNA polymers. They're made out of a lot of different stuff. So that's just a recognition right there. And compared to the eukaryote has a 50S, a 5S, a 28S. I don't know why I said 50S. I was trying to say a 5S, a 28S, 18S, 46 different peptides for the large one. And you can just see, if I were you, I'd just pause this and I would draw this down on my notebook, and I would memorize these, because later on we kind of understand that these different are, for example, that the 23 is responsible. 23 is the RNA, ribosomal RNA 23S is responsible for catalytic activities, and this just gets a lot easier if you memorize it. So here's a question to ask. What are the roles of the RNA in reference to the mechanism of translation? So we have already discussed this before. I just said it right now, strangely. The rRNA 23S and the RNA 28S are ribozyme functions. And ribozymes are enzymes that are catalytic enzymes. And so this is important to remember, 23S and the 28S. And notice the 23S is inside the prokaryote, and the 28S is inside the eukaryote. And these will help the amino acid tRNA linkage we talked about before. So the amino acids on the tRNA is going to help, it's going to help form the new peptide bonds from this tRNA and the amino acids that are bonded together.